All right, guys. This week we've been talking about Mendel's dominant and recessive inheritance patterns, and sometimes it just doesn't do that. So what we're going to do is I'm going to do this little video talking about the exceptions. Okay, the first exception actually looks a lot like what they used to call blended inheritance. Okay, the idea is that if you have a red flower and a white flower, when they cross, they actually have pink flowers, which is exactly the blending idea. However, it's not exactly blending in the way that we would have thought it, because once these hybrids that are now pink, once they have kids, if you self-pollinate them, you actually come back with the red and the white, and some of those kids have pink. So the idea is that the alleles for red and the alleles for white stay distinct, but if you have two red alleles for the gene, you have red flowers. If you have two white alleles, then you have white, and if you have one of each, then you have pink. Now notice what we did here with these symbols. Instead of a big R and a little r or something like that, what we've done is we've said, okay, we're going to use C, the capital C, for the color. And then there's the color red and the color white. So what we've done is we've added a superscript to describe the color rather than big and little letters, because that would assume that there was an actual dominant and a recessive. Okay, so when you do Punnett squares with this, you use your C with the subscript as a single letter, a single symbol. Okay, so you got two CRs here, two CWs here. Obviously, they can only make CR gametes, so they would join up with the CW gametes from this plant. You end up with the hybrid. All right, when they go to make kids, then the sperm, you could have one CR and one CW, and then the eggs, one CR, one CW. So you end up with kids, 1/4 of which would have red flowers, 1/4 of which would have white, and then 50% pink. So that is called incomplete dominance, because red's not dominant over white and white is not dominant over red. They just influence each other so that it meets in the middle. All right, now there's another type of inheritance that is called codominance, which is actually quite similar in idea. You can actually, just by looking at the picture, you can see that we use the symbols with the superscripts. The difference is that the two alleles don't affect each other. They're both seen. Okay, so you got one cow that has two white color genes. You got one cow that has two brown color genes. Well, when they have a kid, the hybrid actually is white with brown splotches. So the white is not hidden and the brown is not hidden, so neither are recessive, but also it's not a lighter brown, it's white with brown. So we call this codominance. They are both expressed. Another place where that's seen is in human blood typing. Okay, so look at this. We have the allele I superscript A. If you got two of those, then you have A-type blood. And what that means is that your red blood cells have these A markers on the outside. They're actually sugar chains, and we'll talk about that more in depth later. In this blood cell, this is somebody with B-type blood because they only have the alleles for B. All right, so they only have the B markers on the outside. Well, in a codominant situation, if you have the A and the B alleles, then you have both markers. So this isn't blending. It's not like an A/B. It's you have A markers and you have B markers. Now notice this guy right over here doesn't have any markers, and that's O-type blood. I'm going to come back to that later. All right, so this cow is expressing codominance, not incomplete dominance, not a mixture, but both. And this cell is expressing codominance, not a mixture between A and B, but both A and B. All right, well, that brings up an interesting question with this guy. Our book calls this example incomplete dominance, and the idea is that if you have a black chicken and a white chicken and you mate, you end up with these blue chickens. Now they're actually gray, but they kind of have this blue tinge to it, so we call them blue. So all the hybrids are this blue. The reality is that this chicken is making both black feathers and white feathers. So if we were to zoom in on this guy, which I cannot do right now, if we were to zoom in on this guy, you would actually see both types of feathers. Of course, as far as the cross is concerned, if you mate between the F1 generations, you would end up having, again, 25% black, 25% white, and then 50% of this blue. So it's a good question. Is this incomplete dominance? Are they blending? Or is this codominance where both of these are being expressed? You can think about that. All right, the third example, so we've talked about incomplete dominance, we've talked about codominance. The third example is multiple alleles. Okay, multiple alleles is when you have one gene but more than two common alleles. So instead of just the big T, little t situation, you actually have three or more alleles that are common in the population. So the example is human blood type. So you got one gene, okay? So one gene determines what blood type you have. But the three common alleles, we've already seen these, the I with the superscript A and the I with superscript B, but there's also a little I, which indicates that it is actually recessive to these two, all right? So if you have one I superscript A allele, that means that you are going to produce these A-type sugar chains. We saw that in the picture prior, and I'll show it to you again. And if you have the IB, then you have B sugar chains. So going back to that picture, there's two ways for you to have A-type blood. You can be homozygous or you can be heterozygous with the little I, because remember the little I is recessive. So the I big A is the only allele that's functioning. All right? Well, likewise, there's two ways to get B. So either of these situations, either of these genotypes gives you B-type blood. Either of these genotypes gives you A-type blood. As we said earlier, if you have A, B, if you have one of each, then you have both. So remember that was codominance. But because we have the little I involved, there's actually a fourth possibility as far as the phenotype of blood typing, and that is O-type blood. And the only way to get O-type blood is if you have both little i's. In other words, you don't have information that says I'm going to put these A markers or the B markers on the blood. All right, that actually, all of this comes into play when you're looking at blood transfusions and things like that. I'll talk about this later, but the idea is that if you have A markers on your blood cells, then you make an antibody that makes it really bad for you to get B-type blood or AB-type blood. Okay, likewise, if you have B-type blood, you got the B markers, but you make antibodies against the A-type blood, so you don't need any of that. So anyway, so that has to do with whether you can receive blood or donate blood and that kind of thing. All right, so incomplete dominance, codominance, multiple alleles, and then lastly, we have what's called polygenic inheritance. In this case, there's more than one gene, not alleles, but more than one gene that affects a particular trait. So in this example is skin color. And the idea is that, and this is a simplified version of the reality, but let's say that there are three genes, A, B, and C, that all affect skin color. And the idea is that the more of the dominant alleles that you have, the darker your skin is. So let's just say that the dominant alleles, say, produce more pigment, and the recessive don't say that. So if you are an individual that has three dominant alleles and three recessive alleles, you would be medium in color. All right, so if we mate two triple heterozygotes, look at all the possibilities for them, okay? Because each one can make 8 different types of sperm or 8 different types of eggs, which gives us a big old Punnett square of 64 squares. But if we look at the 64 squares, there's only one of them that did not receive any dominant alleles, therefore that's the lightest possible combination, and there was only one that received all 6 of the dominant alleles, so that's the darkest possible combination. But if we just count up the number of the number of dominant alleles, you have that list down here. There's 1 out of the 64 that has 0. There are 6 that have 1, 15 that have 2, 20 that have 3, and then back to 15, and then 6, and then 1. So you end up with a curve like this, which means if you mate these two parents, most of the kids will be kind of in the middle, and some will be on the extreme. Now, obviously, with humans, we're not going to have 64 kids to count, but over a big population size, this is what you would see, the kind of the middle group dominating and fewer on the edges. So those are our 4 exceptions to Mendel's regular dominant recessive. I hope you learned something and that you can do the homework for tonight. Thank you.